historical milestones, that is the cell theory and advancements in the plant tissue culture. So we need to know about the various types of the cells. So there are different types of the cells are there in the nature. So these cells are broadly categorized into two types, that is a prokaryotic type of the cells and eukaryotic type of the cells. Again, these eukaryotic type of the cells are classified, or we can say that the cells from the fungi, the cells from the animals and cells from the plants. So there are some basic differences among these cells. The size of the cells vary from a few micrometers to few centimeters, for example, hen's egg. So there are many different type of the cells are there. So who discovered these cells? So if we talk about that, so Robert Hooke was the first to observe these cells under the microscope. He developed microscope and he observed the cork under the microscope, and he observed small regular boxes under these and he named it as a cell. So credit goes to the Robert Hooke for the discovery of the cell. Antony van Leeuwenhoek, during the same time period, he also developed a microscope for the observation, and he observed different type of the cells under the microscope. Particularly, he observed protozoal cells under the microscope, and he was able to draw these protozoic cells on the paper. So these are the early events regarding the discovery of the cell. So further events what we say are also important. So the discovery of the cell is a very important event in the development of the new field what we say the plant tissue culture. So we'll talk about that. German botanist Jacob Schellen was the one of the pioneer worker who stated that all the cells or all the plants and plant tissues are composed of the cells. So his vision regarding this, or his concept regarding this plant cells, plant tissues are made up of the cells is very important. So Robert Hooke and Leeuwenhoek, they proposed or they showed that cells, cork is made up of the cells, or many different type of the organisms are made up of the cells. And after that, in 1939, Jacob Schellen proposed that plants are made up of the cells. So this is one of the important event during this development of the plant tissue culture. So he proposed this statement that plant cells make the body of the plant. Then next person in this field who was again German, and he belonged to the field of the zoology, Theodore Stern, in the same time, he also proposed one concept that all the living what we say the animals are there, so they are also made up of the cells. So cells make the body of organisms. In case of the plants by Schellen, and in case of the animals, Stern suggested that animal tissues are composed of the cells, or the animal body is made up of the cells. So this is one of the important concept which was suggested by these two German scientists. So Rudolph Verchow, he proposed one more concept and he completed cell theory. So we are talking about the cell, how the cell looks like, how it develops, and regarding that Virchow's concept was very important by stating that or from his study he concluded that the new cells are formed from pre-existing cells. So this concept was very important. What Schellen and Stern stated that the organisms are made up of the cells. It may be plant or it may be animal. But Virchow added one more thing, that so how the new cells are formed. So new cells are not coming from anywhere. God is not going to make that cells. So new cells are produced from the pre-existing cells, means already the cell is there and that cell is going to be divided and the new cell is formed. As earlier we have seen, so male gamete is a cell, female gamete is another cell, and these cells will fuse, and by the fusion of that cell, so zygote is formed, so the new cell is formed, or what we say the fusion product is there. So during the sexual reproduction, that zygote will develop into the embryo, and that embryo will give rise the new plant. So this is true in case of the plants, this is true in case of the animals too, because they all are made up of the cells. In case of the plants, we have also seen that we can take the twig or part of the plant and we can grow it into the whole plant. It is possible by means of the cutting or by means of the layering or by means of the other methods, or using the propagules or the small parts of the plant. It is possible to grow the new plant, means there is already cell which is going to grow. So this concept is very important, and this concept was extended as a cell theory by Verchow, and his a famous statement or his a famous conclusion, omnis cellula e cellula, means new cells are formed from the pre-existing cells. So here is a cell theory. So when we talk about the cell theory, whatever Shellen and Sean has stated, all living things are made up of the cells. We can conclude from their statements that all living things are made up of cells. And another thing, that cell is a structural and functional unit of all living organisms or all living things, and all cells, after the addition or extension of this cell theory by Verchow, all cells come from pre-existing cells by means of the cell division. So whenever the new cells are added, so we see the growth. So for example, when we sow the seed and from that seed new plant is formed, so there is an addition of the new cells, and by that means there is a growth or addition of the cell, means the cell are dividing again and again, and they are dividing in a particular way. So see, the zygote was a single cell and that converted into the multicellular cell. Again, the development is in a particular manner. So cell is going to be divided and in a particular manner, and because of that it is able to produce, so that a zygote cell of the plant is able to produce root, stem and leaves and flowers too. So this development is in a particular manner. So that we need to understand. So after that, during 1900, Gottlieb Haberland, again German botanist, he realized that plants can be grown from the small part of the plants, or what we say the vegetative propagules of the plant as well as the seed. So how they grow there in the environment. So when we sow the seed to plant, when there are favorable conditions like that, sufficient moisture is available, the seed will start to grow. Means that inside that seed, whatever the embryo is there, it is going to grow, and that is going to develop into the new plant. As well as other, he was a keen observer, and because of that he also observed many things like that. What we say the vegetative parts can be also utilized for the same purpose. And so he thought that whether it is possible to grow the cells under the control conditions, and he tried to do so. And during that he used media which was containing the organic compounds, and he tried to grow. And during his experiments during 1890s, he found that so the microorganisms or contaminants are going to compete with that plant material he want to grow under the such a controlled conditions. And because of that, he was able to understand the importance of asepsis, that is the aseptic conditions or the environment free from different type of the microorganisms or contaminants. And because of that, he did further experiments, he modified his technique, and he was able to maintain the cells for several days. So he cultured cells on a Knopf salt solution during 1898. In his culture, the cells were able to synthesize starch. It is because the cells were able to maintain themselves on that medium where the sufficient nutrients were available, and aseptic conditions were also maintained, and cells increased in size. However, he failed to induce the cell division, and this failure was because of he was not knowing about that. So different plant growth hormones which are required for the cell growth and development or cell division. So at that time, none of the hormone was discovered. Later on, so there was a discovery of these plant growth hormones. Despite of his failures, he proposed new field, that is what we say the plant tissue culture. So see how is the history of the plant tissue culture. We also see many different type of the plants growing there, with even plant leaf can grow into some plants gives the new bud, and that from the leaf, and that may grow into the new plant. Roots may give rise the new plant. So we regularly observe this, and despite of that, so we never thought, or our forefathers never thought about this thing, the plants can be grown in this manner. But he proposed new field, that is a plant tissue culture, and because of that, Haberland is known as a father of plant tissue culture. So his work in the field of the tissue culture, particularly as a pioneer worker in the field of the plant tissue culture, is very important. He shown one or the new pathway in the field of the plant sciences for the culturing or for the growing the new plants, and because of that he is known as a father of plant tissue culture.